Hello, everyone, and welcome to today's webinar. Go with the flow for rapid viscosity results with Honeybun. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Unchained Labs. To learn more about our sponsor, please visit unchainedlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Kevin Lance, PhD, Director of Biologics and Gene Therapy Marketing at Unchained Labs. Kevin, you may now begin your presentation.
Kevin. I'm here to show you how you can go with the flow with Honeybun, the most rapid viscometer out there. In the world of antibodies and other biologics, the big trend is to move towards high concentrations. But those higher concentrations come hand in hand with the problem of high viscosity. When you're developing a drug for injection, viscosity is the biggest factor telling you if your sample will struggle with syringeability or injectability. In the worlds of antibody engineering and formulation, viscosity raises the alarm when your antibody is prone to sticky self-interactions or if your formulation isn't going to cut it. In process development, high viscosity can lead to issues with filtration or fill-finish processes. But the problem is, getting viscosity data is too slow, so no one is getting as much viscosity data as they need. Old school instruments use too much volume and only run one sample at a time. Even modern tech that just needs microliters of sample has complicated workflows that require hours of hands-on time and add to that with lots of upkeep from clogging and cleaning. Honeybun is the only rapid viscosity system built to solve these problems. Honeybun can take 10 samples at a time and in just a few minutes get viscosity data. It does this using only 35 microliters for each sample, and samples up to 10 centipoise will run in less than 1 minute, and Honeybun can run a full range from 0.5 to 150 centipoise. To prep your sample only takes minutes since all you have to do is pipette it into the consumable and work through the quickest software steps you've ever seen. How does Honeybun do this? Well, it all starts with its consumable, the bun. Each bun can run from 1 to 10 samples, and any channels that aren't used can be saved for later. To prep for a run, simply add in 35 microliters of a sample into an input well, and that's it. Gone are the days of filling syringes or cleaning expensive chips that are prone to clogging, since these buns are disposable. The bun's secret ingredient is the microfluidic channel connecting the input well and the output well. The microfluidic channel has two sections, the resistance channel marked by an R, and the measurement channel marked by the M. Inside of Honeybun, compressed gas pressure pushes the sample through the resistance channel. The pressure applied, we call that delta P. Notice how the resistance channel is much smaller than the measurement channel. That's what allows the channel to have high resistance to flow in the resistance section, but then the larger measurement channel means that the flow can be tracked over a few minutes by a simple camera. In the measurement channel, a camera is used to see the leading edge of fluid, and then velocity and flow rate, Q, can be calculated. Viscosity is determined by dividing the pressure applied by the Honeybun and the flow rate through the microfluidic channel combined with the length, width, and height geometry of the resistance channel. To load the bun into the Honeybun, all it takes is a simple push. The loading mechanism works just like loading a memory card into a computer, or if you're old enough, loading a floppy disk into also a computer. Then the Honeybun software is designed to get you to viscosity answers as fast as possible. From the Honeybun home screen, all that's really needed is to name the experiment and pick which channels where you're running samples, and that's it. If you want, you can click Start. Or you can name your experiment samples and adjust temperature, but to run the software, you only really need 3 clicks of the mouse. After you load up the sample and hit Start, you'll get to watch as the samples flow through the bun so you can keep an eye on which samples are flowing fastest. All the data is automatically exported afterwards as an Excel file, an HTML file, and an image showing how each channel ran. And if you want, you can save the video too. Here's exactly what a run looks like inside of Honeybun as it applies pressure to push sample through the bun's microfluidic channels. This is shown in real time. How fast samples move through each channel is related to the viscosity. Here we're looking at water, a 2 centipoise standard and a 6 centipoise standard at positions 1, 2, and 3, and that's repeated twice more on this bun. You can see in the overlays how the software is tracking the leading edge of the sample so it can later calculate viscosity. Here we're looking at an actual video of running a sample on Honeybun to show that it really is that simple. All you have to do is tell the instrument where you're running the sample, pipette 35 microliters into the bun. So here I'm running a sample of glycerol and water with a little bit of blue food coloring so we can see it better on the camera. And then push the bun into the Honeybun, and we're ready to go back to the software and hit start. You notice as soon as that bun was loaded into Honeybun, then the live video popped up, and the first thing that happens after you hit start is a short temperature incubation to make sure that the bun and your sample are correctly at the temperature you set. Most viscosity experiments are going to be run either at 20 or 25 degrees Celsius, but Honeybun can control sample temperature between 10 and 45 degrees. After the incubation, what we'll see here on the left is that sample is going to run through the resistance channel and then start being tracked by the software, just like we saw in the earlier video on the last slide. So this whole run, because it's on a sample that is less than 10 centipoise, takes about 1 minute, and that includes the 20-second incubation that we saw. When the sample reaches that line of circles, then it's completed its run. All of the velocity data has been tracked, and the instrument's going to calculate viscosity from that pressure versus flow rate curve. All we do then is again pop the bun back out of the instrument, and we're done. That's a complete viscosity read done in about a minute on a single sample. But if we wanted to run 10 samples at a time, it would take the same amount of time to run that bun. Okay, when we do run 10 samples at a time, the results that we'll see look something like this. So here we're looking at 10 different IgG concentrations formulated in PBS. We're looking at averages with error bars showing standard deviation. You can see as concentration increases, then viscosity also increases for our IgG sample. And in this case, our standard deviation is actually coming from Honeybun's ability to run multiple shear rates in a single run. So each data point is really the combination of many calculated viscosities. So what that means is Honeybun will start the pressure low and increase the pressure high during each run so that sample starts slowly and runs faster. And we're going to take the average and standard deviation to show that average viscosity here. Because Honeybun is taking samples through that pressure ramp, that also means that we get a lot of different shear rate data for each one of those samples. So at the same time that all this viscosity data was gathered, Honeybun also applies that low to high pressure ramp on these antibody samples, which means they run slower, which is lower shear rates, or faster, which is higher shear rates. And we get an understanding of this, if each and every sample is showing Newtonian or non-Newtonian behavior. The viscosity of Newtonian samples will be flat across different shear rates, which is what we're seeing. And non-Newtonian shear thinning or thickening samples will show dramatic change at different shear rates. When you run samples on a bun, you know that each bun is served up with precision. So to show this off, IgG from 0 to 75 milligram per milliliter was measured in triplicate, but each replicate is actually measured on a different bun. Honeybun's precision across all those samples has a less than 2% coefficient of variation, which is great. And the specification for Honeybun on this kind of experiment would be around 3% CV. If we want to compare that type of experiment between Honeybun and old school cone and plate rheometers, it's a little bit more convenient to do it on high concentration glycerol water samples so that we see much higher centipoise, which is more ideal for reading samples on a cone and plate instrument. And you can see that here the results agree within 1 to 4% between Honeybun and our cone and plate instrument, where they're both getting the same viscosity results at the controlled 30 degree Celsius temperature. But importantly, if they agree, you're still going to want to run samples on Honeybun, because even with these high concentration, high viscosity samples, Honeybun's running those in about 10 minutes total, and you get through 10 samples at a time in those 10 minutes. Meanwhile, the cone and plate experiment, because you're running one at a time samples, you're going to take about 120 minutes to get through all 10 of those samples. If we ran these samples on a microfluidic system, where there are other microfluidic viscosity systems out there, running those 10 samples takes about 28 minutes. And because Honeybun can run things at any temperature, you can get your viscosity data at that range of temperatures as well. So here we're showing a really quick and easy experiment where we've taken a single concentration of our favorite IgG sample and run it at 4 different temperatures to understand how viscosity changes across temperature. But where Honeybun really shines is the ability to do both temperature control and high throughput experiments at a very precise, with very precise viscosity data. So here we're looking at temperature control between 10 and 25 degrees Celsius at 5 different concentrations of IgG, where we're running samples in triplicate at each one of these data points. So across all of these results, we're still seeing great precision, where we've shown the highest concentration coldest sample still shows a 0.8% CV. On any other device, this would be a massive undertaking, but on Honeybun, we're actually done in less than a few hours, where Honeybun's results are exactly needed to pinpoint the trend in viscosity versus concentration for this antibody, and understanding the trend for temperature for this antibody. If we want to take a look at another case study on Honeybun, we can have, say, this therapeutic antibody where we want to target which concentration we want to use for our injectable formulation. So if we go to really, really high concentrations, in other words, what's going to be ideal for what we can actually inject? Well, the first part of that problem is understanding the concentration that our antibody solution is at. So for that, we're going to use our stunner instrument, which is going to confirm the concentration with a minimal volume of sample. Stunner is going to bring together quantification by UV vis with the ability to gather size and polydispersity data from DLS. Those are great because they're both label-free, fast methods. Those reads are going to be done on the same 2 microliter sample for up to 96 samples at a time, and that makes Stunner ideal for determining antibody concentration and to check for any aggregation at the same time on the same sample. So here we see exactly what that looks like. I'm using water with the fluorescein dye just so you can visualize it well on camera here. You load in 2 microliters of sample into each one of our microfluidic wells on Stunner, and that will wick into the sample and be prepared to be read inside of each of those microfluidic circuits. Coming out of that, you'll get both UV vis information, where we're going to look at the absorbance peak at 280 nanometers, and you'll get DLS information where we understand the distribution of sizes that our antibody sample has. For this case study, if we run our sample and analyze it at A260, we're confirming our concentrations between 36 and 150 milligram per milliliter. We also ran a 4th sample, but that did exceed Stunner's 40 centipoise viscosity limit, so we did extrapolate for that higher, roughly 300 milligram per mil sample. And then by DLS, also read at the same time on Stunner, we're seeing PDI values between about 0.15 and 0.25, which indicates a little bit of aggregation is going on, but nothing crazy where we don't have massive aggregates present. We'll also note that with the graph shown on the right, the correct size values does actually need the viscosity data from Honeybun fed back into the system so that we know the right size for things based on the viscosity that they have in solution. So if we take 4 different antibody concentrations and our buffer solution as well and feed those through Honeybun, the results we get look like this, where we have our buffer sample, and then as we're adding more and more antibodies into solution, we see viscosity goes higher and higher, all the way up to our 300 milligram per milliliter sample. And we're crossing our injectability viscosity limit, which is between 20 and 25 centipoise, somewhere between our 150 and 300 milligram per mil concentration. So that gives us the ability to start focusing more on where between those two antibody concentrations we need to be to exactly stay to maximize our concentration, but stay under our viscosity limit. And if we want to take advantage of Honeybun and look at the shear rate data that's gathered at the same time across those samples, we can look at that data and see that all of these samples are behaving with Newtonian flow, so they're nice and flat across the shear rates measured. So key part of all of that viscosity workflow to understand concentration and viscosity behavior is Honeybun, because with Honeybun you can finally get all the viscosity data as you want, as fast as you want it, and nothing will be stopping you from getting your viscosity groove on. With that, I'd love to take any questions you have for me.
Well, thank you, Kevin, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, are the buns reusable?
So each channel in a bun is one-time use, but you can use one channel on one day and come back the next day to use the others. So that means you never have to worry about chips clogging or cleaning up afterwards, since you can just use each of these channels one time and move on.
Perfect. Great. Thank you. Our next question here asks, what is shear rate good for?
Okay. So it's helpful to understand if you have Newtonian or non-Newtonian samples. If you're trying to inject a non-Newtonian sample, for example, the viscosity could increase or decrease as your injection force is applied. So basically, you're trying to push this sample faster. It's going under higher shear rates. How does viscosity change? Sometimes it stays constant, sometimes it increases, sometimes it decreases. So looking at a range of shear rates helps to characterize that behavior.
Great. Our next question asks, how long does it take for 1 CP or 150 CP samples?
So at 1 centipoise, a Honeybun read is about a minute long. You saw in the video, it takes about 45 seconds plus a 20-minute or 20-second incubation time. And at 150 centipoise, that's about 10 minutes total, somewhere in the 8 to 10 minute range.
Great, thank you. Next question here. You mentioned Honeybun runs based on compressed gas. What kind of lab gas supply is needed?
So Honeybun works great with nitrogen from a house supply or from a tank, since it doesn't consume much volume. So even a tank will last a while. It also works great with compressed air. And if using compressed air, that air just needs to meet a certain quality spec, that the gas is dust, free of dust, water, and particles, like a clean, dry air type of supply.
Great. Next question here. Does the measurement require a dye in the sample?
No, nothing extra is needed to a sample. It's really quick to just pipette your microliters out of a tube and add it into the Honeybun. A lot of videos I'll show something with like a blue or green dye added. That's just because the buns are transparent and it can be hard to see what's going on without a dye.
Great. Well, we're going to go ahead and wrap up here. Kevin, I want to thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Unchained Labs, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.